All right. The first and most common form of cellular transport. This is intracellular transport. Intra means inside of, okay, or within the cell, right? Intracellular transport is almost entirely accomplished by the process of diffusion. Diffusion happens naturally. It's free. The cell doesn't need to expend any energy to use this process. All things naturally diffuse down their concentration gradient. That means from an area where they're highly concentrated to an area of low concentration, which essentially means they're almost always going to end up where you need them, because where you need them, you don't have very much. And so it's naturally going to go from where it's produced, where it would be in high concentration, to places that have used it up, right? So that's great. It's a free process that generally does the job we want. There's a couple of drawbacks to diffusion. First off, it's slow. Okay, so as you can see, getting something for free is you get what you pay for. Right? You've probably noticed that, right? Like if you buy an app for your phone, right? There's the free version and then there's the paid version. And the free version is okay. It generally does what it's supposed to, but the paid version almost always works a lot better because it's got better features and whatever else. Well, the cell is using the free version. It generally gets the job done. But it doesn't guarantee that the material being produced is going to end up where it's needed. And it's not targeted. So if A is where the stuff is being produced and B is where it's needed, okay, there isn't a tight beam of materials moving from A to B. Diffusion doesn't work that way. Okay? Diffusion is more like the shotgun approach. Okay? That is that the material spreads out, okay, out of the barrel. It generally moves. If it's moving from high concentration to low concentration, it's probably in low concentration in a whole bunch of places. Okay, so as it moves away, it spreads out. You can kind of see that in this diagram here, okay, with this ink that's going into this pool. So we got right here at the spout, obviously it's very dark, it's very concentrated, but as it moves away and naturally diffuses, it's getting diluted. Okay, it's diffusing away, becoming more diffuse. Okay, it's kind of like if somebody farts, okay? Because this is diffusion. It's just diffusion through the air. If you're right next to the person who farts, the smell is really, really strong. Okay? If you're a little further away from the person who farts, the smell isn't as bad because it's diffused into more volume before it gets to you. Eventually, the fart diffuses everywhere. Everyone gets to savor it. And then it kind of is just gone. Okay? Because it diffuses to the point where it's no longer detectable. Right? It's diffused into such a large volume that we don't even notice it. Well, if A is where this stuff is produced and B is where it needs to go, the farther A is from B, the less of that material is actually going to arrive at B. So diffusion is okay as long as the distances are small, which in a cell they are, because a cell is microscopic, obviously. Okay? But it's also not guaranteed that all of it is going to get there. Okay? But again, the cell doesn't need to expend any energy to use this process, and, well, money's important. Okay? Our energy is important in this case, so we don't need to worry about expending a lot of energy. We're going to use that process, and cells typically use that process almost entirely, okay, for moving stuff within the cell. Now, that also has an implication for cell size, which we'll talk about later. Okay? Cells then can only become so big before diffusion becomes ineffective, because diffusion is slow and it doesn't guarantee it's going to get stuff where it's supposed to go. Okay, you wouldn't use Canada Post if they couldn't guarantee. Okay, maybe that's a bad example. Sometimes things get lost in the mail. I mean, they're pretty good about getting. I mean, I think I've had like 2 things lost in the mail my whole life. So, they're pretty good about getting things to where they're supposed to go. Okay. So you use them, right? If you want to send something to somebody else, you use the mail. You don't go outside and go, all right, I'm just going to throw this crap in the air and hope it gets there, okay? Because that's too far, right? It's not going to work. Well, it's not going to work because that's a stupid example, but okay. Obviously, you send it to somebody using a courier that says, I'm going to take it from A to B, and I guarantee that from A to B is where it's going to get. Okay? You don't take it to somebody who says, yeah, I'll throw it in the air and hopefully the wind will take it where it's supposed to go. Okay? But if it was a short distance, like let's say you only needed to give something to your neighbor, well, you could throw it over the fence. Okay? I mean, it'll probably get into your neighbor's yard that way, right? As long as you're close, that's okay. But if you're far away, use Canada Post or UPS or whatever, okay? Because they're going to guarantee that it gets there. You probably can't throw it far enough and rely on the wind. Okay? All right, so diffusion is known as passive transport. It requires no energy from the cell, okay? Simple rule of diffusion, okay, is a substance will diffuse from where it is more concentrated to where it is less concentrated, which generally means it's going to get where it's needed, generally. Okay? The drawbacks, it's slow, and not effective over large distances. And of course, I wasn't knocking Canada Post. Canada Post is very reliable. Okay. I hope no one takes offense to that. Okay. Questions on that? Okay. So transport across the membrane. So diffusion is how the cell moves stuff within the cell. Osmosis is movement across the cell membrane. So from outside to inside and from inside to outside. All right? Transport across a membrane has to work differently than transport within the cell, because now we're crossing a barrier that's designed to keep things in or out. Okay. Now the cell membrane is what we call semi-permeable, which means some things can get through it and some things can't. Water is one of the things that can easily move across the membrane. And that's important because water is crucial for basically everything to work. Right? So osmosis is the passive movement of water, okay, across the membrane. We should add that part. Okay? Osmosis is the passive transport of water across the membrane. Here's the key. Osmosis only moves water. No other material moves by osmosis. Only water. Right? So if a question on an exam is talking about calcium or it's talking about glucose or anything other than water, it's not using osmosis. Okay, everybody follow me there? Now, the reason osmosis happens is because your cell wants to keep the concentration of salts and other solutes equal inside and outside. So osmosis moves water across the membrane because it's easier to move water than it is to move salts, okay, or glucose or any other material. So it moves water across the membrane to keep the concentrations the same. So if there's a lot of salt inside the cell and not very much outside of the cell, water will move into the cell and dilute the salt in the cell so that it's the same concentration as outside. If there's lots of salt outside, water diffuses through the membrane out of the cell. Okay? That's why if you're ever stranded in the middle of the ocean, you don't drink the ocean water. Also, the ocean is the world's toilet. Don't drink the ocean water. Okay? Everything eventually gets to the ocean. Right? But also it's full of salt, and that will kill you. Right? If you drink the salt water, the concentration of salt in your tissues and your blood goes up. Your body doesn't like that. And your cells have no control over osmosis. It's passive. It's a natural process that they cannot do anything about. So if there's salt in your blood more than normal, water is pulled by osmosis out of your cells and into the bloodstream. And then your kidney detects that the blood has more water in it than normal. So what do the kidneys do? They pull the water out. Okay? And you pee out a whole bunch of water that you really needed. Now how do you feel? Better because you went pee, but you feel thirsty, okay? Because now you've gotten rid of a whole bunch of water. So what do? You drink more ocean water, which just continues to exacerbate the problem, okay? You continue consuming more and more salt because it's making you thirsty, okay? And it's making you thirsty because it's pulling water out of your cells. You're actually better off in the first few days to drink your own urine than you are to drink the salt water. Keep that in mind if you're ever stranded on the ocean. Yeah. That's smart and disgusting all at the same time. You get delusional because you're getting dehydrated. Yeah, it all has to do with dehydration. You get so dehydrated that you just become like. Has anyone ever been like really, really badly dehydrated? Okay, like if you're getting mildly dehydrated, you start getting like a headache. You'll start to feel kind of tired and grouchy. You get muscle cramps. Okay, if you start to get dehydrated. If you get beyond that, then you start hallucinating. Yeah, it's generally really bad if your electrolytes get really out of balance and your water level gets low. All right, so transport of water across the membrane to balance salt concentrations. Okay, so across the membrane, and we'll just write in here to balance salt concentrations. That's what osmosis does. It doesn't move the salt, it moves the water. Okay, so in this bottom picture here, okay, we've got a normal cell under normal conditions. A normal cell under normal conditions has water moving in and out at the same rate. Okay, water's always moving in, water's always moving out, but it's always equal. If I put a cell into what are called hypertonic solution, or hypertonic conditions, what I've done is essentially put the cell in a salty solution. Water will be drawn out of the cell by osmosis, and the cell will shrivel up and die. Okay, so what happens if you put salt on a slug? Okay, the slug shrivels up because the slug doesn't have skin like we do. Its epidermis is much different, and osmosis can happen through it. So the water just comes out to balance the salt, and the slug shrivels up. Okay, if you're in hypotonic situation, that means I have a cell and it's essentially in like distilled water, so there are no salts. Okay, then water rushes into the cell to try and make the concentrations equal because there's way more salt in the cell than there is outside. In fact, there's no salt outside, so is there ever going to be a way to get those equal? No, so water rushes into the cell until the cell bursts. Okay, then the concentrations are equal because the cell is burst. Okay, but it'll just continue to happen. The cell will just get bigger and bigger and bigger until the membrane fails. Biggest cell, I think, is, it's a relative of the amoeba. It's called a slime mold, and they can actually be quite large. They can be like about 2 millimeters or so. Yeah, pretty big. Yeah, but I mean that doesn't sound big, but that's big for a cell, and that's the upper limit of where diffusion can work properly. Right? Okay, so. So does this sort of make sense here? Okay, in this situation, I think you actually do this lab in Bio 20 AP, where you've got a U-shaped kind of test tube, and there's a membrane in the middle. It's essentially like a sausage casing, and you have like distilled water or very low concentration of salt water on one side, and then highly concentrated on the other. And over the course of like only maybe an hour or so, the water level on the low concentration side goes down, and the water level on the other side goes up because water diffuses across that membrane, because it's made of cells, across that membrane to make the solutions equal in concentration. Okay? So it's a process that happens naturally. Cells can't control it. All right, transport within and across the membrane by active transport. Okay, active transport is just like its name would imply, active. It is not a free process. The cell invests energy in this process. And in this case, you get what you pay for. Active transport transports material very quickly across the membrane. Okay? And it does that using carrier proteins. Make sure you make note of the use of carrier proteins and energy. Okay? Carrier proteins are like gates that lock. Okay? When the cell is using active transport, the energy it uses opens and closes the gates. Okay? Essentially what it's doing is changing the shape of these proteins rapidly. So the protein changes shape so that it is open to the outside, and whatever it is the cell is trying to transport is essentially drawn in, and then the cell quickly snaps shut, or the protein snaps shut, and the material is essentially pumped into the cell. Right? So active transport is the pumping, okay, of a molecule against its gradient. That means it's going to an area of more concentration. All the other ones, we're seeking a balance. Active transport creates imbalance by using energy. All right? It's pumping water uphill. It's moving material into the cell where there's already lots of it. Okay? It wouldn't move that way naturally. Energy has to be invested in it to do it. Awesome. Yeah, they'll work against each other, but remember, they're not nearly as fast. Active transport can go way faster. So yeah, if you shut down active transport, eventually the other processes would create a balance again, but at least for the short term, this is what works. Your nerve cells and your muscle cells primarily use this. It's how your muscles can clench and relax. In order to relax, they've got to pump ions out. And to clench, they've got to pump ions in. Okay? And that has to be done quickly, so they use active transport. Okay? All right, so just highlight that, guys, highlight that one thing there, and you're good. Okay. All right, so the three methods that we've just talked about all involve moving materials that are small or that are dissolved in water, so they're in solution already. Right? We're going to move on now to talking about larger particles that the membrane actually has to change shape in order to absorb. Okay, so we're going to be looking at that next. All right, transport out of the cell. If we want to move something out of the cell, that's exocytosis. Okay, exocytosis is controlled by the Golgi apparatus. Okay, so the Golgi, which we talked about the other day, makes these vesicles, makes these bubbles that contain something. Okay, those vesicles move to the membrane, they merge with the membrane, and whatever was inside pops out. Okay, so if you had like something in a Ziploc bag in your freezer that you were going to have for dinner, you would open the Ziploc bag and kind of turn it inside out in order to push whatever was in there out, because it would be frozen to it, right? So you can push it inside out, okay, and make it go out of the cell. Well, that's what's going on here. I've got a Ziploc bag, okay, this thing here. I'm moving it to the membrane. When it touches the membrane, it's made of the same material as the membrane. It just becomes part of the membrane, and whatever was inside pops out. Okay? That's used to do two things: get rid of wastes or secrete hormones. So the hormones produced, okay, within the cell in the smooth ER can be transported either by the smooth ER to the membrane in a vesicle or by the Golgi to the membrane in a vesicle. Okay? Either way, the process is mechanically the same. Okay? So exocytosis is movement out in a vesicle. Right? It's not like active transport. It isn't active transport. It's the physical change in the membrane. Okay? All right. Questions on how that works? Okay, so the Golgi is involved and the membrane actually changes shape. Now, endocytosis. There's three different kinds of endocytosis. They all involve a mechanical change in the membrane. Okay, the same mechanical change that went on for exocytosis, except now in reverse. All right. So here's an amoeba. Okay, you were looking at this last week. Okay, in order to ingest food, what the amoeba does is it just envelops food particles. Okay, it just kind of goes all the way around them, puts its membrane all the way around, and then essentially it's created a vesicle. Okay, we call that a vacuole or a food vacuole if it's full of food. Okay, but it's essentially just a bubble made out of membrane that's going to move now and merge with a peroxisome and then become a lysosome and start the digestion process. Okay, we call that process phagocytosis. Okay, and it's just the physical enveloping of a food particle. Right? So there's phagocytosis, which is cell eating. It's ingesting a solid particle. Pinocytosis, which is cell drinking. It actually just envelops fluid. Cells only do that if they're really horribly dehydrated. Okay? And receptor mediated endocytosis. That's when a hormone or something like that touches a receptor on the membrane and the cell quickly absorbs it and gets it inside. Okay? It's only doing this with molecules or particles that are too big to move through the membrane. Okay? In order to get into the cell, they have to be enveloped by the membrane and taken in that way. They can't go through the membrane. Okay? Everybody follow me there? All right. So phagocytosis, the cell engulfs a particle by wrapping a pseudopod, which is an extension of the membrane, around it and packaging it within a membrane-enclosed sac, large enough to be called a vacuole. Right? This is how your white blood cells kill bacteria. Okay? They eat them. They go around and they just envelop them like that, and then they digest them inside. Okay? And that's how your body gets rid of infections. No, bacterial infections anyway. If it's a viral infection, it absorbs and destroys the cell that was infected instead, okay? Which is why you feel so wiped when you have a virus. It's because your immune system is actually attacking your own cells in order to kill off viruses. All right, so cell engulfs a particle. Okay. You can summarize it with just those terms there. Okay, we don't like talk much about these processes because they're not like true cellular transport. They're more like They're more like how the cell sustains itself by eating and drinking. Okay, so you don't often have questions on these other than maybe a multiple choice question. All right, Pinocytosis, the cell gulps droplets of water, okay, extracellular fluid, okay, into vesicles, so it just needs some fluid, or there's lots of electrolytes in there, okay, things like that, and it would need to get them in a little more quickly than osmosis would do it, or active transport would do it. Okay, usually when this is going on, active transport is also going on and trying to move things in, but under even a light microscope, you can see this process happening. Okay, so this is the membrane, okay, right here. And you can see that it's pinching in and making those vesicles. All right, so whatever it is going into those vesicles and being taken into the cell. Okay, and again, both pinocytosis and phagocytosis are not substance specific. Okay, you can see that when it made this particle, it got lots of the green dots, which is probably what it was after, but it also took in a purple triangle, okay, which is probably not what it was after. It just took in whatever was in that bit of fluid that was against the membrane at that particular time. So is this possibly a high-risk behavior? Could be, right? I mean, you're desperately in need of something. You might accidentally also get some things you didn't want. Okay? Hopefully your cellular machinery can take care of those things. All right. Receptor mediated endocytosis. This is how your cell takes in chemical messengers that the body sends to it. Okay? All over your membrane, and I showed you this the other day, okay, are receptor molecules. They're like little antennas. There's a different antenna for every chemical signal that can be received by your cell. So it doesn't work like the antenna in your car. Your car has one antenna, and it can pick up every radio station as long as you tune the radio the right way. Okay? A cell has an antenna for every radio station. Okay, it's a little different, right? The whole membrane is covered with these receptors, and each receptor has a different shape. That shape can hold the molecule it's supposed to hold, right? So they're like pieces to a puzzle. You can't jam the wrong thing into the wrong receptor. It'll only react if the right molecule hits it. When that molecule hits it, the membrane immediately engulfs it. Okay, same process mechanically. The membrane changes shape and makes a vesicle. Okay, that vesicle will hold all of the chemical signals that were touching the receptors at that time, right? So you can see the process starting to happen here, right? You can see there's these proteins that are on the surface of the membrane, and they're all holding. Actually, you can see it really well right here. Okay, you can see the shape of this thing here. I'll erase this in a sec, and then you can see the receptor molecule sitting on top of it. Okay, So it's actually, you can actually see it pretty well. And you can see here, within the vesicle, a few seconds later, right, that it's almost ready to kind of pinch that off and become a fully enclosed vesicle. And every one of those coat proteins has got a circle sitting on top of it. That would be the molecule it was supposed to absorb. So your cell can absorb this stuff really fast. If it's something like adrenaline, that's what needs to happen, right? You don't want to be waiting, 5, 6, 10 seconds for your body to go, oh, right, you're scared. That grizzly bear is right in front of you. Hang on, we're getting the adrenaline to you. Just relax, right? That's not going to result in your survival of that situation. It's got to not only get that stuff out, but it's also got to absorb it quickly, right? And that's usually what happens. Okay, so RME is very specific. Okay, there's proteins, receptor proteins, specific receptor sites. Okay, substances bind to those, or well, you don't need to know that they're called ligands. Okay, they're usually proteins like hormones that the cell needs. So RME is specific, and it's taking in proteins like hormones. Okay, mechanically still the same. The membrane changes shape. All right, I want you guys to answer those questions. Some of them you'll have to think about, and other ones are kind of straight out of the notes. Right, so give you a few minutes on those. Okay, write down the answers. I think the questions are actually in your notes package, right? So just answer them somewhere, and then I'll go over them here in a few minutes. All right, color blindness. Color blindness is genetic. Okay, it occurs almost always in men as opposed to in women. The reason for that, it's on the X chromosome. Men only have one copy of that. Okay, males all have an X and a Y chromosome, whereas women have two X's. Okay, so because women have two X's, they are much less likely to have the gene for color blindness on both of them. Whereas with men, you only have one copy. If you have the gene for color blindness, you're going to express it. Okay, the reason you are colorblind is because your cells have an error in their DNA. And that DNA doesn't, or that error doesn't allow your cells to produce a protein that you're supposed to have in the cones, that is the color receiving cells in your eye, okay, that allow them to send neural impulses to your brain telling you you're receiving certain wavelengths of light, which makes you unable to tell the difference between certain colors or certain shades of colors, right? How many of you have taken your driver's test where you've had to do the color blindness thing or gone to the optometrist? They make you look at like this jumble of dots and they ask if you can see the number inside the dots. Anyone had that test? Okay, so that's the color blindness test. If you can see the number, you're not colorblind. If you can't see the number, then you are. And you can tell whether you're red-green colorblind, which is the most common, or whether you're just certain shades of red or green that you can't see. Okay, some people, very rare, are basically entirely colorblind. Mr. Van Derve is entirely colorblind. Okay, we actually played a joke on him a number of years ago. We all came into school, and there were actually two of us that were wearing like exactly the same outfit. Mr. Van Derve was wearing something similar, but a different color. And so we went up to him and went, look, how did that happen? All of us are wearing exactly the same clothes. And he went around telling everyone, this is crazy, we're all wearing the same clothes. And everyone's like, Jerry, you're wearing a purple shirt. They're wearing green shirts. See, we're kind of mean to each other in the science department, but Mr. Van Derve can't see colors, okay? Like basically he can see, he knows, like you say, which one's purple, and he's like, that's kind of, and it's not. Like he can't see that. But he knows, like when he comes up to a stoplight, whether the light is red or green. How does he know that if he can't see colors? No, don't rely on that. Don't rely on whether the other cars are moving. You ever notice red is either always at the top or always on the left? There's a reason for that, okay? There's a reason why stoplights are always set up with red on top or red on the left. Is because that way if people are colorblind, they know the light is red, even if they don't know the light is red, okay? Everyone follow me there? Okay, so color blindness is an error in your DNA. It has nothing to do with your cells being able to make proteins. If your cells weren't able to make proteins, being colorblind would be the least of your worries, okay? And in fact, if you have the genetic, and there are a few genetic disorders where that can't happen, basically a woman wouldn't even know she was pregnant if the fetus had that kind of a genetic error. because the fetus wouldn't survive long enough for the cells to even divide. Okay, it just wouldn't work. So yeah, that's not something that would ever be expressed, inability to make proteins. If a cell can't make proteins, it just dies. Okay, so this is unable to make certain proteins, right? And you can survive being colorblind, right? It's not going to hurt you unless you make a big mistake about something being the wrong color, and that's somehow fatal. But okay, that doesn't happen too often. All right. So does that make sense? The problem with color blindness is that it's genetic. It's the same thing with being diabetic. Okay, that section of your DNA that's supposed to code for insulin doesn't code for insulin. It makes something else that doesn't work, and then as a result, your cells don't absorb sugar from the blood properly. Okay, so lots of those types of things are genetic disorders. Number 2, why is it important for organelles to have their own membranes? So each one can have its own proper environment for its function. What was the big long word we used for that? Started with C. I'm sure someone can look it up. First slide. Not the title slide, the first slide. Compartmentalization. I just saw in Jack's notes he even had it highlighted. So compartmentalization, you might want to write that beside your answer there. That's compartmentalization. And we said that's super important because it allows each process to have the right environment to be more efficient. Okay, if a lysosome's membrane ruptured, what might happen? This is a roundabout way of asking what the lysosome does. No, that's the smooth ER. It would digest the cell. Okay, what's inside the lysosomes? Digestive enzymes and food. So if the lysosomes ruptured, all those digestive enzymes would get out, and suddenly your cell would be food, and they would digest at least parts of the cell, if not enough to kill the cell. All right, and number 4, what problems might a cell have if all the ER was removed? Yes, I know that's what you meant. Okay, well, you wouldn't be able to detoxify drugs or other poisons. What else wouldn't you be able to do? You wouldn't be able to make hormones. What else? Wouldn't be able to transport proteins. What else? Smooth ER makes these. Membranes, right. You wouldn't be able to repair your membranes because the smooth ER makes lipids, and lipids are what make membranes. So okay, you'd have a lot of problems if all the ER was removed from your cell. Well, I mean, you'd have a lot of problems if any organelle, for that matter, was removed from the cell, but okay, that one in particular. Again, that's a roundabout way of asking, what does the smooth ER do? Right? And guys, generally, in biology, that's how we ask those kind of questions. We don't just go, what does the smooth ER do, and make you like regurgitate it. We make you think a little bit, okay, about what it does, okay, as a job. Okay, tomorrow you have a quiz. It'll be a cell diagram where you'll be asked to label and give the function of the structures. I will post that later on today. Okay, have a look at it tonight and go over that stuff in your notes so that you're ready for that for tomorrow. Okay, tomorrow we will be doing a lab, okay, the cell anatomy lab. So you'll have the Chromebooks at the start, and I'll go through the lab and do all of that pre-lab stuff. Then we'll go across and look at the slides that we're going to be looking at for this one. This time we're going to be looking for specific structures within the cell. Okay, we're going to be looking at a plant cell, an animal cell, and a couple of other types of cells. Okay, and we're going to see what structures do they have in common. What does that tell us about the basic functions of the organism and the role that those structures play? Are you going to see all the structures in your diagram? Like that cell diagram we labeled on Friday. Are you going to see all of them? There's no chance. Okay, you're not going to see them all. You're going to see the main ones. Okay, you'll see the nucleus and the nucleolus. And if you can see the nucleus, then you can see the nuclear membrane because you know where the outside of the nucleus is. Okay, if you can see where the cell is, you can see the cell membrane because you know where the cell begins and ends. Right? Things like that are things that are easy to label. Cytoplasm can always be labeled because there'll be a blank spot in the cell that just begs for a label. Okay, you'll probably be able to see chloroplasts in the plant cell. You might be able to see the Golgi, okay, in cells. Sometimes that's visible. Sometimes the rough ER is visible around the nucleus. Okay, if it looks like your nucleus has a pointy beard, Okay? You are seeing the rough ER at the bottom of the nucleus, or the top, depending on the orientation. But it looks like there's this furry stuff coming off of the nucleus. That's the rough ER. Okay? Those are kinds of things that you want to look for when you're looking in the cell. It's not going to look exactly like it does in your diagram. You have to, I hate to say this, you have to use your imagination a little bit, okay, when you're looking at the cell and you're deciding what things to label. Okay? Be sure, though, all right? If you're looking at something going, I'm not sure, maybe that's ribosomes. First off, it's not, because you can't see ribosomes. They weren't discovered until the electron microscope was developed, so don't label those. You can't see them. Only label what you can see and what you're reasonably certain of. Okay? Now, if you label nothing, then obviously that's bad. Okay? Label some things, but label the things you can see. Okay, questions there? All right.